Thank you, everyone, for joining us today for today's Seek Space webinar. Today's webinar from Dr. Nicholas Papadopoulos is about the early detection of cancer challenges and opportunities. Before we introduce our speaker, I want to quickly review a few webinar logistics. For this webinar, all lines will be in listen-only mode. You may submit questions at any time using the Q&A or chat panel and selecting all panelists. And Tabitha Harrison will ask it on your behalf during the Q&A session. You may need to activate the appropriate panel using the menu option found at the bottom of your screen, as displayed here. Note that closed captioning is available as a link in the chat box, and this webinar is being recorded. And with that, I'm going to pass the microphone to Fred Schumacher, who will introduce our speaker.
Thank you. Dr. Nicholas Papadopoulos is a professor of oncology and pathology at the Johns Hopkins University School of Medicine. His research focuses on cancer biology, genetics, and diagnostics, and he is internationally known as a co-discoverer of the genetic basis of the predisposition to hereditary non-polyposis colon cancer, one of the most common hereditary forms of cancer. In this webinar, Dr. Papadopoulos will present on liquid biopsies and challenges and opportunities in early detection of cancer. I will pass it off over to you.
Thank you. Thank you. Good afternoon, and thank you for the opportunity. Let me share my screen. I hope you can see that. Okay, so I hope everybody can see the screen. So today, today, what I would like to do is take you through our thinking and the efforts that we put in trying to develop tests for the early detection of cancer. So seems that I don't have control of it. Here we go. So application of DNA sequencing in cancer has been around for a long time, not only as a research tool, but as you can see here, as a means to try to derive information that is useful for the management of individuals. Germline, sequencing of germline can provide information about hereditary cancers or risk assessment for the individuals. Sequencing the tissue can provide information for prognosis or therapy selection. And recently, circulating tumor DNA has at least a promise to actually deliver a number of different applications, like early detection and screening that I'm going to talk about today, minimal residual disease detection, monitoring, response response prediction, and monitoring resistance for individuals that they have undergone therapies. So today, I will focus on early detection only from all these applications, and specifically in trying to develop tests for the early detection of cancer in average risk individuals. And I like this cartoon. I kind of wrote underneath what it says here because it might be blurry. I'd have been here sooner if it hadn't been for early detection. This old man says here, and then some older people waiting in line to get into paradise anyway. And really, that's part of the idea to actually prolong of early detection. The goal is to prolong survival and decrease of deaths due to cancer. But as we move forward, although I'm going to talk about the technical challenges, biological challenges, and so on, we cannot forget that as we develop those tests, we cannot forget that there are many challenges to early detection. So there are social challenges, as I call them. I think in contrast to cardiovascular disease, in cancer, we are more reactive to the disease than proactive. There are psychological issues, and how we communicate potential results to individuals is very important. Telling somebody that he or she has cancer, it can create, especially with the false result, it can create anxiety, depression, and a lot of psychological issues, perhaps. Also, negative results, they can be mislead individuals thinking that, oh, now I have no risk for cancer. So as we develop tests and communicate the results, those things are important to keep in our mind. The performance of the test, of course, is important. We're going to talk about that. Nothing is going to be 100%, neither the sensitivity or the specificity. So we care about overdiagnosis, which really leads to the issue, which is overtreatment and unnecessary follow-ups. On the other hand, we miss a lot of cancer. So we do have also the issue of underdiagnosis. And tests that are designed to be applied to the larger population, they have to be economical and they have to be practical in that they can be easily accessible to individuals. So the goal really of early detection is to identify what we call screen-detected cancers. That's cancers in individuals that actually do not have symptoms or they do not know at least that they have cancer. And that, again, is not enough. What we really want to find is the cancers that matter. I know this is not good terminology, but with this, I mean cancers that we can do something about it, we can manage the individuals, and really those are the cancers that they need to be detected in order to follow some intervention with the intent to cure or prolong survival. The earlier we detect the cancers, it's better. This is data from SEER database, and this just as an example, ovarian cancer. Most of the ovarian cancer detected very late when the survival is suboptimal. But that means really that we missed them when they were regional, and I think that we missed them when they were localized. The minority of the cases are the ones that are detected with localized cancer, and those are the ones that actually there's the highest chance for survival and better chance for treating successfully these individuals. This is not specific to ovarian cancer, it's for every cancer, really. And this is from an older paper. This is from stomach cancer or gastric cancers. These lines, one represents a male and the other one female. But as you can see, again, the earlier we detect them, the higher the chance for survival. And now even current therapies, even like immunotherapies, have a better chance of successful outcome when the number of cells or the tumor burden is less, and usually less cells or a low tumor burden is associated again with earlier stages of cancer. So as we move forward, there are several also considerations. We need to select biomarkers for the test based on what is the intended use. And I think through the examples that are used, this will become more clear. We need to define what is a true positive for the test and also define what is the target population. Not every test is going to fit for every clinical application. So blood-based liquid biopsy, it does provide an opportunity, and this is because it's easily accessible medium, it's blood, it's minimally invasive to collect it, and it should be able to reach many demographics. It's easier or more practical to collect blood from somebody than to send in for some more invasive or more expensive procedure. Also, another opportunity in blood-based liquid biopsy is that instead of having a test for a single site, a single tumor type, you can actually develop tests that they can detect multiple cancers in one blood draw. Some of the advantages for that is instead of single sites that are excluded certain cancers by definition, while it's more inclusive for the multi-cancer. The other thing is for the standard of care screening that now is recommended, only 20% of, at least in the United States, of individuals that are up to date with all of the tests that are recommended, for example, mammography and colonoscopy, and if you're a smoker, low dose CT and so on. So most of them, they actually are up to date with one or two of the recommended tests. So in multi-cancer cases, actually, we have also some that we call aggregate prevalence, and I'm going to go over that briefly in another slide. So detecting multiple cancers, it also can be more efficient. So here's also a hypothetical example, screening for pancreatic cancer, average risk individuals, let's say that you have, again, hypothetically a test that is 99.5% specific, 60% sensitive. So I had this hypothetical example to make a point, and what I was saying is that the 500 false positives here that there will be for a test that if you develop only for pancreatic cancer, because the specificity is 99.5 and it's a rare cancer, you have a blood test to detect multiple cancers. Actually, that 500 is going to be cumulatively for all potentially all the tumor types that this test can detect. So the false positives are shared between the different tumor types, if you wish, and that is an advantage of multi-cancer screening for blood tests. Then I was saying, how can we distinguish cancer from non-cancer? There are a number of different biomarkers that have been used and being used, and tests are being developed that are utilizing them. And a lot of them, of course, sequencing of DNA is how we determine the signal, like DNA mutations, copy number, DNA methylation, fragmentomics, chromosome positioning, and even for the cases that people develop biomarkers that are based on transcripts or exosomes. Exosomes contain nucleic acids. Again, that's how we detect the signal. So there is common challenges to the sequencing for all these biomarkers. Now, technical challenges developing those tests. The signal from cancer is really small compared to the signal that comes from non-cancer cells. And it's the proverbial needle in the haystack that a lot of us in the field we use to describe this problem. So this is an older publication, but they start noticing that mutant fragments per 5 mL, so that's what this graph is, from different stages. You can see the average, it's lower. So low stage cancers or early stage cancers, they have fewer mutant fragments that they come from the tumor DNA, but that is on average. You can also appreciate the range, even in stage 4, the range of the number of molecules. So not every tumor, even from the same stage or same type, behaves the same in terms of the amount of DNA that is shed into the circulation. So this is from a study of stage 1-2 pancreatic cancer. And again, it's ctDNA molecules per ml. This is 66 cases. Each black dot is one case. And you can see the red line. This is 2 molecules per ml. So that is some of the issues that we have to keep in mind as we develop those tests. They have to be very sensitive to try to deal with this small number of signal, the small number of molecules that they carry the signal we want to detect. And the other challenge is the accumulation of errors that they can lead to false positives. So you start with initial circulating tumor DNA fragment, then you have to do some type of target enrichment, either it's PCR or capture or making a library for sequencing. Then you have to sequence it, and that is, again, another chance for accumulating errors. And then depends on what the output is. There could be in silico errors too that they can mislead and result in false positives. So some of the advances to address those challenges, those technical challenges, there were 2 main ones, I think. And one is what we call now digital genomics. And that it was first Bernd Vogelstein and Ken Kirzler in 1999 in a PNAS publication that described digital PCR, it was called at the time. And the idea is that instead of using the analog to try to, the red one, let's say, is the molecule you want to detect that comes from cancer. The rest of them are wild type or a signal that comes from non-cancer cells. Of course, if you use an assay that looks at all these molecules at the same time, probably you're not going to have the sensitivity to detect this one molecule that is the signal from cancer. So the digital approaches, they really have basically assayed every fragment individually. So that's why there are individual vehicles here. This way, there are two advantages of that. One, obviously, you can, the signal from the mutant molecule, let's say here, or the molecule with the methylation from the, or whatever your biomarker is, it's actually equal now to any other one that comes from non-cancer cells, because each molecule is going to be assayed individually. And the other one is it gives you also the ability to enumerate and find out how many molecules of the circulating tumor DNA is present in the circulation. So the second advance was there are a number of what we call error-reducing methods that have been developed, and that is to distinguish genetic and epigenetic changes that are present in the starting material, in the fragments that are present in the blood, versus similar changes that develop during processing. This is the error, the accumulation of errors that I referred to a couple of slides ago. And for that, there are many laboratories and investigators that have developed. We have developed one, actually, Isaac Kinde, back 2011 from our group, an MD PhD student, developed something that we called safe sequencing system. And the advantages of that is, or the features is as follows. Let's say you have fragments in the blood of the DNA. One of them has a mutation, that's the red star there. And then you go through the manipulations. The first step, although, is to try to assign a unique identifier, which is a molecular barcode, to each fragment that is present in the blood. So one gets the pink, the other gets the yellow, and so on. In this case, the mutant got the light green, wherever the color is there. And then you go through all the manipulations, either PCR or sequencing, and so on and so on. And in the end, the output of the sequencer, you bin those according to this unique identifier. And the assumption here is that, and it's a safe assumption, that all these molecules really came from that one first molecule that it was present in, in the stage before the manipulation of the DNA. So most of them will have no change. I mean, most of those molecules will be wild type. Then those will be considered errors. It's clear when you see it this way that actually those were errors that if we didn't have this system to bin everything under the same unique identifier, those could have been called signal from the cancer. Either it could be a mutation or methylation or some other change that you use as a biomarker and become a false positive result. So this is what we will call a mutation, the ones that are actually all of the molecules or most of the molecules, they have the daughter, I guess, molecules, they have the same mutation. So that is sensitive for most of the applications that we have because really it decreased the error rate significantly. But can this be even better? Can we improve it more? And there is one case that this type of approach doesn't take into account, and that is what if actually the DNA that was present in your plasma, in the plasma that you wanted to analyze in the blood, it actually already had some damage, that it had nothing to do with the cancer, right? It was a damage that was in the DNA for some reason. And the solution to that is to be able to actually sequence both strands of the same fragment of DNA individually. And there are a number of attempts. We did this in the past. There are some other. This is just an example of the first, I guess, two attempts to do this, at least in blood. And they work, but they had also significant challenges, and that is loss of fragments. And as we discussed, losing fragments that are very rare, that has issues, creates issues with sensitivity, and also target enrichment. Like, for example, sometimes you need to capture 2 or 3 times. That was one of the things that the investigators, the Smith et al., found out. So he had some issues that actually then we thought that we can try to address them. And Josh Cohen, another MD PhD student, just recently published well, actually now it's published, sorry, I say it here in press, a paper that addresses that in the following way. So we discussed that this would not fool us, even without the sequencing of the Watson and the Crick strand, and we would never call this a mutation. But this is what I showed you earlier, that it gave us confidence that this maybe it's a real mutation, but it's only in one of the two strands. That's probably a mutation or that came, that is there due to some damage of the DNA, something that has nothing to do. It was only in one of the two strands, other in the Watson and the Crick. So this technique, this method now, what it does is we call mutations, bona fide mutations, only those that there are in both strands. So that will be now the advance of that and the real mutations. So here are a couple examples. This is P53 mutations. Those are positions in the P53 gene for this region that this mutation is in. And the different colors, what they show is different admixture of the mutant DNA to the DNA that is from an individual that has no mutations in P53. And you can see the noise. The mutation, this mutation is here, the other mutation is there, and you can see the noise, although, again, this is 0.01, 0.01% is right here. So the noise, it is, manageable, but a lot of times we want to go very, to be very sensitive. And this is really what happens when you actually use a technology that looks at mutations in both strands, eliminates this the background, if you wish, and the artificial mutations. That, what it does is increases the sensitivity, and it increases sensitivity because now we can drop the limit of detection even lower than the 0.01% for in these cases. So it really helps us to do that. This is how it is with barcodes only in one strand. Again, the error rate is about 2 to 3, 10 to the 5th, and the other error rate now with both strands goes down to 2 to 3, 10 to the 7th. And that makes a very sensitive technology to try to detect mutations in this case. Now, there are biological challenges too that are harder to address because they are not just artifacts and technologies. They are there because of biology. There is clonal proliferation, for example. There's chip and mutations can be present as people of older age. Benign neoplasia can have methylation, for example, that has nothing to do, or comorbidities, methylation or mutations, or other biomarkers can be artificially increased that they have nothing to do with cancer, but they have to do something with the comorbidities. And also with this very sensitive technology, the one that I described, we start coming down to what we call biological limits of detection, which is there are non-clonal mutations. I mean, there are always some mutations or changes in the DNA, perhaps in one cell, but we start detecting those, that will be confusing too. So the other biological, if you will, challenges, and I mentioned that earlier, the amount of circulating free DNA from different tumor types varies, and it's not exactly clear why, but the fact is that it does vary, and that has effect in sensitivity. Like this is an example between liver and colorectal cancer. I saw you in the previous slide that in the study, liver actually circulating free DNA from people with liver cancer, it's a lot more than from people with colorectal cancer. And you can see the effect in sensitivity here in this particular study. It can detect almost all of the liver cancers, but not all of the colorectal cancers. So what can we really detect then? If we put this together, we can detect a lot of different tumor types in blood, not all of them. There is hard time for some tumor types is difficult to detect them for whatever reason that we're not going to address in this talk. And also, again, to remind you that it's even harder for stage 1 compared to stage 2 or 3. But also what we cannot detect. Again, biology plays a role. There are some cancers that we call them non-setting, whatever that means. Really, it's not a real terminology, but the fact is I show you that not every tumor from the same tumor type or stage, the amount of DNA that is in circulation it's not the same. You can predict. Are those all indolent? Not clear yet. There are studies that they try to do that. And then detection of precancerous lesions is not going to be sensitive. If we have a hard time detecting stage 1 cancers, for example, probably it's going to be harder to detect precancerous lesions. Of course, some of those can have some positive, if you wish, outcome in terms of overdiagnosis, since a lot of indolent cancers or some precancerous lesions maybe we shouldn't be detected. But the studies have not been done to really demonstrate that. The last test is like, what is the test performance? And there is always a play between sensitivity and specificity when develop a test. Then that depends on the prevalence. The predicted values will depend on the prevalence of the disease. So, for example, for specificity in our mind is of paramount importance in screening. Cancer screening, with that I mean average individuals that they do not know that they have cancer or they don't have symptoms. Only a very small percent, about 1%, depends on the age of the group that you screen, will actually have cancer. So specificity has to be very high. Now, in a diagnostic setting, sensitivity, I will argue that it's very important because that is the goal of a diagnostic, to try to give information about what the disease is. And in high-risk individuals, you have this interplay, again, depending on the prevalence of what is the cancer that you try to detect, but you can actually give a little bit from the specificity in order to increase sensitivity, since these individuals are probably being followed. But the goal overall is to increase sensitivity without affecting specificity or still keeping specificity very high. And one way, the way we're thinking about it, is actually a combination of markers and biomarkers, all these markers that I showed you earlier. And Josh Cohen developed a test we called CancerSeq. It was a combination of circulating tumor DNA and proteins in this case, where we detect cancer, and we use the information also from proteins and some of the mutations to actually localize the cancer, or at least give an idea to where the cancer may lay in the individual. That was a case control study, had 1,005 cancer patients from 8 different tumor types and 812 healthy controls. There were no metastatic cancers. So that was by design. All those were cancers, even the stage 3, they were resectable cancers. About 69% were stage 1 and 2. And we used a small panel of regions in the genome, the usual suspects for genes that are commonly mutated, but a very small, less than 2,000 base pairs. And the idea of that is something that I mentioned earlier, keeping it practical, keeping it, having a panel that actually it can be manageable and not very expensive. And then we added proteins. Again, those are, if you wish, proteins that are usual suspects. They were elevated in cancer based on scientific literature and other studies, but they have not been used for detection of early detection of cancer. And we use them in very high thresholds. And I'm going to use one example to show you what we mean with that. That is CA99. The clinical value that is considered important is 37 units per ml. That actually, in a set of 221 stage 1 and stage 2 pancreatic cancers, 182 controls, it gave decent sensitivity and you could say decent specificity, but this is not what we like for a screening test. The 55 units per ml, it came from a study from a different lab, Kim et al., trying to avoid pancreatitis, I think. And what we use for our studies is a really high cutoff of 92 units per ml. And you can see it, the sensitivity takes a hit, but we get good specificity. Now here it says 100% in this study. Of course, it's not going to be 100% when you do thousands of individuals, but here that's the idea to how we use those. And if we use multiple proteins plus the the circulating tumor DNA, we hope that actually this decrease in sensitivity per marker is going to be picked up by other biomarkers that are in the panel. So, for example, I mean, these combinations, they can also target different biological things, like circulating tumor DNA really comes from dead cells that they die in some way. Protein markers, they may be coming from dead cells too, or being present in exosomes, but also they're secreted from live cells. So kind of try to balance to what biomarker can give you. For example, mutations, they give you the genetic for the cancer, while fragmentomics, methylation, can tell you the state of the cell, the cancer cell that is there. So I think a combination of these markers is going to complement them each other, and it's going to create, in our mind, a test that is going to be hopefully useful. So briefly, the results of the CancerSeq, again, you can see at sensitivity greater than 99%, but still, again, the issue that I showed you earlier, not every test is not as sensitive for every single tumor type. So how can we increase this, improve this? Methylation markers, fragmentomics, already I mentioned in a couple of slides ago that including the other markers or testing them in combinations to try to see what actually is going to help us increase the sensitivity. But keep in mind, again, specificity has to remain at greater than 99%. So one example I'll show you, I would like to share with you is that aneuploidy again is that again is detected, as I mentioned earlier, by sequencing circulating tumor DNA. And that started, it's a technology that we use where a single primer can actually amplify multiple regions in the genome. And that is because the primers target lineage, they target the repeats in the human genome. And then we tile it, and depending on how they tile in the genome, you can have amplifications or you can have deletions. Chris Duval, an instructor in our group, actually took this to a different level. With a single primer can amplify 350,000 genomic regions in all 39 chromosomal arms of the human genome. As it does that, also identifies SNPs. So there are multiple readouts from this one PCR pair technology where you can detect aneuploidy or allelic imbalance, focal amplifications. Also, there are size differences, and also we can use it for sample identification. That's coming from the information that we receive from the SNPs. So when we looked at the CancerSeq cohort, this is the samples that I saw you with the 8 different tumor types. You can see the sensitivity here for liver for mutations. Then this is for aneuploidy. So in some cases, there is no help. In some cases, actually, one analyte is better than the other. Like in those two cases, esophageal or ovarian cancer, ovarian mutations give better sensitivity, and so on. And then when we actually combine those with also the proteins, what was the CancerSeq, in every case, there is an improvement. Sometimes it's not as high of an improvement in sensitivity, but overall, the combination of those three actually provide great sensitivity. And again, it's a specificity kept at 99 or higher. So there are a number of examples like that from our group, from other groups that they use different other multiple analytes of fragmentomics, methylation, aneuploidy, and all of them look very promising. And that's why I think there is a hope for that those biomarkers may work. But we think that the way to test those eventually is in a prospective interventional study. The goal is to target the population, to do the study in the target population. And if the target population is average risk individuals, that means that usually they're going to have a smaller size cancers, they're going to have an earlier stage disease. And also, if you have in observational studies, you can't really report results. So it's harder to figure out what is the harm that is done using this test. So we set up a prospective interventional trial, a study. Sorry, it wasn't really a trial, but prospective interventional study that we call DETECT-A to answer. It was more of a feasibility study. We wanted to see, can we detect cancers in a prospective manner in individuals that they don't know that they have cancer, or they didn't have previously cancer, or they didn't have any symptoms, and can we intervene with intent to cure, and also how does this play with standard of care screening? Can these two tests actually help each other to detect more individuals with early stage cancer? And finally, something, of course, very important, what is the safety of this test? What are the follow-up tests and procedures that we follow? So the whole study was set up trying to have a lot of, I'm not going to go through details, but a lot of safety, if you wish, checks, because we haven't really done this before with liquid biopsy tests. So the blood test, it was an earlier version of CancerSeek. Sometimes that's how things work. The study started earlier, and now we have some of it didn't include the algorithms that they were developed in the 2018 paper, or the aneuploidy, or the increased sensitivity method detection for mutations that I saw you that it was just published. But again, it's a feasibility study. It didn't include DNA, circulating tumor DNA and proteins. We eliminated CHIP by testing the BAFI code in the test positive cases. That's a way to eliminate mutations that are not associated with cancer. And again, it was a small the panel that I told you earlier, so it can be efficient and cost effective. The next step is, where is the tumor? I told you that when we first developed CancerSeek, we had an algorithm to try to detect where the tumor is localized, or to get some information. Methylation and fragmentomics seem to actually doing a pretty decent job in doing that. And once you get a signal, let's say colon cancer, the follow-up probably is going to be colonoscopies. There will be some type of imaging or procedure that it comes after that. But if that turns out to be negative and still you're very confident that there is signal, that could be confusing and actually can set up a cycle where, okay, it's not in colon, what do you do next? Either you don't do anything, or maybe it's something else and you follow some other type of imaging or so before, some treatment decision if needed. So we decided in this study actually to use a PET-CT for a tumor of origin. It is a FDA cleared test, and it's an orthogonal confirmation of blood test, and we like that. That is a totally different modality to validate the result that we had for the blood test. As I'll show you, actually, it helped in reducing some unnecessary follow-up after the PET-CT. It was uniform and accurate localization, uniform in that every individual in the study that had positive signal in the blood had the same follow-up procedure, at least that before a diagnosis was in place. And it provided really information that it was actionable. So it wasn't saying, oh, maybe you have lung cancer. That is what a molecular tissue of origin will have. He said it was on the left or the right, for example. It gave us information about the size. Was it metastasis? Information that it wasn't just the information that maybe there is tumor in some organ. It provided more information to these clinicians. So this study was designed as following having the blood test, no follow-up. If it's positive, there will be follow-up after review of a multidisciplinary committee. The results of the PET-CT, they were reviewed again, and then another decision point was made for intervention or nothing to do. The population was about 10,000 women, 65 to 75. And although this enriches for ovarian cancer and other cancers, older people have more cancers, they also, this population is enriched in comorbidities. So although the study was not designed to have special, certain groups with certain comorbidities, but really trying to address a number of comorbidities that they will be present in this age. So we thought that that was better test for the specificity of CancerSeek. The only exclusion criteria was current or previous known cancer. and all of them were enrolled in Geisinger with access who had to EMRs, and also minimize the follow-up. So from the 10,006 women, there were 9,911 actually that took the test. 95 were excluded because they turned out actually either they wanted to be in the study, some of them, but they did have symptoms, or they had some history of cancer that it was discovered at the screening. So those were the 9,911 individuals out of the 10,000 or so. So the first one, can we detect prospectively in these 10,000 individuals? There were a number of different cancer types that were detected. The PPV of the blood test was 19%. With imaging, it was 41%. And in this study, it was 10 cancer types that they were detected. That doesn't mean that's the only 10 that they can be detected. That was the ones that was in this population. And a little bit more than half of them were detected by mutations. Intervention after the detection, they were about 64% of them, they were localized or regional. And they were, based on that, there were 12 surgeries with intent to cure in different sites, as they are highlighted here. In the question about how this can be incorporated in the standard of care screening, these individuals had the standard of care screening. The green are the cancers that they were identified by the standard of care. So there were some colon cancer, breast cancer, and lung for people that had, they were smokers and they had low dose CT. There were no overlap. So really the blood test doubled the cancers that they were detected with standard of care. And we wanted to see if these people would be deterred of following up with the standard of care. So in a fraction of these individuals, we looked their compliance with mammography. There were women, all of them. So, and we didn't see a difference between the ones that before and after the test that they took the participation that they detect A, which was again good news for us as we developed the test. In terms of safety and follow-ups, of course, the vast majority of the individuals had nothing because they had negative tests. There were 101 PET CTs, and I told you earlier that actually PET CT stopped significant number of follow-ups based on what the multidisciplinary committee saw, how the mass was looking in the individuals and other imaging radiographic criteria that they were used. So from those, there were 22 that they had some type of minimal invasive procedure and 3 surgeries, and the 3 surgeries turned out to be here with the square. They turned out to be actually necessary. One was large colonic polyp with high-grade dysplasia. The other one was an in situ carcinoma, and the other one turned out to be a 10-centimeter ovarian lesion that was a mucinous adenoma. So there were no serious adverse events. So I mentioned earlier, there were 96 cancers in these 10,000 individuals, 25 detected with standard of care, 27 by blood test screening, no overlap between the two. So the total, when I mentioned the double, the number detected, it's 52. 31 of the cancers detected by the blood, they actually do not have a standard of care screening. So I think we can detect the answer in the 4 questions, cancers, and there is hope that these things could work. It can enable treatment with intent to cure. It can also be additive and complementary to the standard of care screening. We don't envision any blood test dethroning some of the standard of care screen that we have now, and it's nice to see that they can work, to be complementary and additive to each other. I mentioned about the PPV with and without aging, and we thought PET-CT worked pretty well, at least in this setting. Next, we still think that we need randomized prospective interventional trials in the target population, need to determine clinical utility, risk benefit, cost effectiveness, and so on. And again, it's concepts, we discussed some of them already. Multi-cancer, some dental cancers are low prevalence, and single site tests will require very large studies. I'll show you some hypothetical examples of that. We still think, at least initially, we need to focus on specificity as we try to increase sensitivity. We need to decrease the time frame for approval. With that, I mean, it can be many, many years of studies, and we need to find out proxy endpoints that actually will tell us if those tests are working, and hopefully with real world evidence post-approval we can see the reduction in mortality. So very briefly, the last couple of minutes, this was for average risk, that as I went through almost all the talk, how you develop these panels. For high risk, the panels and the biomarkers may be different. They have to be relevant to the target cancers. You're not going to use, for example, mutations or methylation sites that are not present or they have not been shown to be present in the cancer type that in high risk individuals. So the panel specific to cancer associated with risk. Still, it could be a blood test, but there are a number of cancer types that the blood test may not turn out to be, the appropriate medium. And for example, it's very hard sometimes to detect glial cancers or cancers of the CSF and in specialized, this is not for screening, obviously, but in some situations, CSF could be the medium, or urine, or saliva, or pap smears. So the way we see it is that the other fluids, as I call them, or the other liquid biopsies, they have their utility too. For example, people with hematuria, I don't think that can use a blood test that is designed for the average risk population. Rather, you can use the same technologies, different panel, targeting what cancers potentially can be detected in urine. Similar idea of detection of ovarian endometrial cancer or detection of head and neck cancer, especially individuals that they have some symptoms. So really our vision is to integrate those tests in routine screening and management of the individuals. Many thanks to, of course, Bert and Ken, and Sibin and Chetan, my colleagues at the Ludwig Cancer Research Institute, and Anne-Marie, very instrumental in those clinical studies that I described, Christian, that develops the algorithms for detecting the signal from cancer in the blood. Ralph is our pathologist and head of pathology, and Elliot, an expert in imaging, and some of our sources for funding. But the big thanks goes to the 10,000 women that they voluntarily participate in the DETECT study, and we learned so much. And the second big thanks goes to all these individuals that work at the Ludwig family, as we call them, that they actually do the studies. And thank you for your attention.
Thank you, Dr. Papadopoulos, for an interesting and comprehensive talk on a very innovative area of research. Unfortunately, due to technical glitches, we don't have much time to address questions. However, I would like to take a few questions from the audience before we wrap up. So you mentioned target populations for intervention studies. Could you speak to that a bit more? So for example, should relevant populations be required for circulating tumor DNA-based detection methods, such as patients with pancreatitis for pancreatic adenocarcinomas?
Yeah, I mean, that's, I think some studies have, I don't know if we can address every single, I assume you're talking about developing a test for the average population, like screen that I talked today. I don't know if we can address every single, potential comorbidity in a separate study. And that's why I think having, you can do, I don't know, we can do the math too, but you will require a few hundred of this comorbidity, a few hundred of the another comorbidity. I think something that we learned from DETECT-A, for example, is that having the target population with whatever comorbidities they may have, it might be sufficient. That doesn't mean that you shouldn't study specific situations like pancreatitis, especially when you actually developing a test, let's say, for pancreatic cancer for high-risk individuals. So that's how I see it. I don't think we can go through every comorbidity and having a separate study for that comorbidity for the average population.
That makes sense. Another question is, how important is it to evaluate survival outcomes? And could you talk about the feasibility of expanding the size and duration of these types of studies to examine outcomes?
Yeah, I think so. That is obviously a controversial in some ways subject. And I mentioned in one of the slides that I think as a community, and I've been in some discussions with that with different people, we need to find a way to not have to wait for 20, 30 years to figure out if nobody's going to do these studies anymore. The I think also we need to learn from history. I think, and you can correct me, but even colonoscopy that now is considered the best screening modality to have for colorectal cancer. It took many, many years in order to be even accepted. I remember when I was developing tests for detecting, the HMPCC genes, BRCA1 and BRCA2, some of the questions that I was like, oh, maybe they are not useful. I think either we think that trying to develop something to detect cancer early, it really helps. Yes, sometimes it could be lead time bias, but if you catch it at a stage where actually you can manage an individual without having risk for his or her life, I think this is something that we need to think it also that way. If we believe in early detection and we have examples of early detection that worked in the past, I think we need to find proxies to actually see that those technologies don't take like 20, 30 years before they get, the acceptance. But this is just one view, right?
Well, I think while we would love to continue talking with you about this area of research, I think we need to wrap up. So I'm going to go ahead and turn it back over to Leah.
Great. Thank you, Tabitha. And thank you, Dr. Papadopoulos, for a fantastic presentation. I'm sorry for the technical difficulties, but I think we still got a great presentation from you and a great discussion. I just want to let everyone know that our next SeqSpace webinar is scheduled for October 12th from 3 to 4 p.m. and will feature a presentation focused on somatic mutations and high-grade endometrial cancers by Dr. Michelle Cote. You can register for this SeqSpace webinar on our webpage now. Thank you, everyone, for joining us today, and I hope you have a great rest of your day and a great evening. Thank you.